Hello, my name is Emily Donnelly, and I'm an undergraduate researcher in Dr. Justin Lakeletter's lab. My current project focuses on using stabilized Huntingtin oligomers to determine their mechanism of lipid membrane binding. Huntington's disease is one of many neurodegenerative diseases in the same class as Alzheimer's disease and Parkinson's disease. These diseases are protein misfolding disorders, which are caused by rearrangements of a protein's conformation to form aggregates or clumps of protein. The protein that is misfolded in Huntington's disease is Huntingtin. The first 17 amino acids of this protein are referred to as NT17. A polyglutamine expansion, or a repeated sequence of the amino acid glutamine in NT17, allows Huntingtin to aggregate into several different forms, which can damage lipid membranes within cells. NT17 affects both aggregate formation and lipid membrane binding, so a possible method of treating Huntington's disease could come from blocking Huntingtin's ability to interact with membranes. Previous data from our lab has shown that the oligomer state of aggregation has the highest affinity for membranes. When the lysine residues in these oligomers are cross-linked with difluorodinitrobenzene, or DF-DNB, they are stabilized. They do not further aggregate into fibrils and do not interact with membranes. In our current project, we attempted to prove that this stabilization was caused by a change in the flexibility of the oligomer's conformation, not a change in the charge of the linked lysine residues. First, Huntingtin mimic peptides known to stabilize oligomers were mutated so that the lysine residues lost their positive charge and became neutral. The residues affected were K6, K9, and K15. 7 combinations of these residues were used: K6, K9, and K15 alone, K6 and 9, K6 and 15, K6, 9, and 15, and regular NT17. Oligomers were allowed to form using these peptides and incubated for 8 hours. Samples were taken at 1, 3, 5, and 8-hour time points. The samples were then analyzed using atomic force microscopy, or AFM, to determine what type of aggregates were formed. PDA, or polydiacetylene, and thioflavin assays were also performed. On the bottom left, there is a diagram of how atomic force microscopy works. A cantilever with a nanoscale tip moves over the surface of the sample, moving up and down over structures on the sample's face. A laser bounces off the cantilever, and its change in position is recorded by a sensor, allowing us to make a sort of topographical map of the sample's surface and determine the shape of any aggregates. The results of the thioflavin assay showed that the cross-linked samples formed fewer fibrils than regular Huntingtin. This can be seen in the graph of the assay, where the dark blue line represents unaltered Huntingtin and the other lines show cross-linked proteins. The results of the PDA assay showed that the samples retained their ability to interact with the membrane. A higher colorimetric response in this assay means more interaction with the membrane, and as you can see, all the samples had at least some interaction with the membrane. The K6, K9, and K6, 9, and 15 samples had slightly higher interaction. Finally, AFM images showed that the samples formed oligomers and did not aggregate further into fibrils. It also showed that they remained stable for the full 8 hours of incubation. The images in the top right show the control, which contains fibrils, and the K6, K9, and K15 samples, which contain oligomers at the 8-hour time point. This experiment showed that neutralizing the charge of the lysine residue stabilized the aggregates into oligomer form without removing membrane activity. Therefore, we can conclude that the loss of membrane activity in the cross-linked oligomers was due to changes in their flexibility, not charge. While these are only preliminary results, we now have more information on the mechanism of oligomer stabilization and can perform further experiments on preventing membrane damage. Thank you for viewing my presentation.